Welcome to the fourth module for Prime Immunology. In the previous chapters, we offered an introduction to the innate, adaptive, and complement systems. Throughout this module, we're going to dive a little more deeply again into the adaptive immune branch, this time to talk more about antibodies. Remember from before that as part of the humoral immune system, B cells differentiate into plasma cells. Plasma cells are effector cells whose job is to produce antibodies, which are also known as immunoglobulins. Also remember from before, antibodies come in all sorts of different shapes and sizes. As a result, specific types of antibodies will predominate during a given infection depending on the timing or particular agent in question. So let's dive in deeper into these types of antibodies. IgM is found in blood and lymph fluid and is the first type of antibody made in response to an infection. It acts to neutralize microbes, toxins, or other antigens, and it can activate complement through the classical pathway. As you can see in this image, IgM is very large and it's a pentamer. IgG antibodies are found in all body fluids and are really important in fighting off bacterial and viral infections through neutralization and opsonization. Similarly to IgM, IgG can activate complement through the classical pathway. Also, IgG antibodies are the only antibody that can cross the placenta in a pregnant woman to help protect her fetus. Let's pause for a moment here. This is a chart showing the general timeline of antibody production during early versus late stages of infection. As we already mentioned, IgM is the first type of antibody that's produced a few weeks after initial exposure to an antigen. Because of this, if you suspect a patient has a certain infection, you can check their plasma for IgM that is specific to the microbe in question. If positive, this suggests that the patient has very recently been exposed to the infection. Conversely, IgG is produced later than IgM. When testing a patient for previous exposure to an infection, or testing to see whether a patient has been vaccinated, you can check whether their plasma contains IgG against the microbe in question. Moving on, IgA antibodies are found in areas of the body such as the nose, breathing passages, and digestive tract. Since they're found in mucosa and mucosal secretions, IgA antibodies protect body surfaces that are exposed to outside foreign substances. Fun fact, while IgG is the most prevalent type of antibody in human plasma, and therefore the most abundant circulating antibody, IgA is actually found in highest numbers overall in the human body. Lastly, IgE antibodies are found in the lungs, skin, and mucous membranes. Through sensitization, they cause the body to react against foreign substances such as pollen and animal dander, and are therefore involved in allergic hypersensitivity reactions. IgE levels are also often elevated in patients battling parasitic infections. Here's the same chart from before, summarizing key basic functions for each of the antibodies we discussed. Keep in mind that antibody function is more detailed and complicated, and this is just an introduction that hits the highlights. But don't worry, you'll soon learn so much more about this in your immunology instructional block. Thanks for your attention.